Okay, welcome to Group 12's Journal Club presentation. Today we're going to be discussing resveratrol PLGA nanoparticles for the treatment of NAFLD, which is non-alcoholic fatty liver disease, and the actual article I've just written down here. Okay, so let's get into it.
So just before we get into the nitty-gritty of the scientific article, I just wanted to give some contextual background information. So non-alcoholic fatty liver disease is the most prevalent chronic liver disease worldwide. The causes of this disease are multifactorial. However, major factors include a high-fat diet, calorific abundance, a sedentary lifestyle, and some papers do discuss a genetic predisposition to contracting the disease. The disease state is strongly associated with obesity, insulin resistance, and metabolic syndromes. So the pathophysiology of this disease occurs in hepatocytes, which are liver cells, and the disease essentially occurs as accumulation of fat, called steatosis, occurs within the cells. So this steatosis leads to oxidative stress, cell inflammation, and cell death. So this cell death is responsible for the formation of liver fibrosis, which is commonly described as scar tissue, and this consequently leads to cirrhosis. So that's where the liver can no longer function correctly. So this pathophysiology is responsible for the progression of liver failure, which is a really serious condition, and where the treatment options are quite limited. Okay, so let's dive into the introduction of the article. Okay, so currently treatment options for NAFLD have shown that the regression of fat cells can be achieved through lifestyle changes, such as having a balanced diet, increasing activity, essentially achieving a calorific restriction. However, pharmacological intervention has shown to be of some benefit in assisting the reversal of this fat accumulation, especially resveratrol. So resveratrol is a naturally non-flavonoid polyphenol phytonutrient. So essentially meaning that it's a chemical compound found in plants, and its essential role is as a primary purpose of helping the plant to thrive off predators, competitors, or pathogens. So resveratrol has specifically been found in grapes, knotweeds, and peanuts, and it's been shown to have some outstanding potential in antioxidant, anti-inflammation, and anti-cancer processes. So in regards to non-alcoholic liver disease, studies have suggested that through targeting SIRT, which is an enzyme responsible in lipid metabolism, and through the lowering of inflammation, that it may be a successful option in the reversal of hepatic steatosis and fibrosis. Although the effectiveness of this pharmacological treatment is still contested in the scientific community, further investigation is required to truly identify its therapeutic effect. However, as the preliminary literature does show some success, it is the aim of this study to look at the limiting factor of bioavailability and essentially have a better idea of the therapeutic outcome of this pharmacological treatment. Okay, so resveratrol's limitation of bioavailability is largely due to its poor aqueous solubility, its chemical instability, and intestinal metabolism. So when an individual takes it, it's essentially not able for the body to actually accumulate this drug. So it is hypothesized that through the intervention of this nanocarrier, the PLGA with polylactic glycolic acid, that the drug will achieve a higher bioavailability and essentially will give more information surrounding this drug's effectiveness. So the function of polylactic glycolic acid is to essentially encapsulate the resveratrol, and through its highly biocompatible and physical strength, allow the protection and the slow release of this resveratrol. So the aim of this experiment was to enhance the stability, solubility, and bioactivity of the resveratrol by optimizing the method of delivery through PLGA nanocarriers in vitro to treat non-alcoholic fatty liver disease.
The methods of this experiment can be broken down into three main sections: liver cell and PLGA nanoparticle preparation, simulating the human body to test the effects of resveratrol PLGA nanoparticles, and the modelling of non-alcoholic fatty liver disease. Part 1. Liver cell and nanoparticle preparation. For this experiment, human cancer liver cells HepG2 was utilised due to their highly prolific properties. These cells were cultured with fetal bovine serum to ensure a high-quality culture was produced and streptomycin to prevent bacterial invasion. They were incubated at 37 degrees Celsius with 5% CO2 to simulate the human body. Once cultured, these cells were subcultured by taking a small portion of the original culture and splitting it into 3 different tubes as replicates. In order to encapsulate resveratrol, PLGA was used. PLGA is a biodegradable copolymer that is used as a highly compatible delivery system for drugs as it has minimal systemic toxicity. Resveratrol was encapsulated in PLGA as a carrier to strengthen its stability, solubility, and pharmacological potential. This was achieved by the following. PLGA was dissolved in dichloromethane and acetone, which was used as a good solvent to form a well-proportioned PLGA solution. Resveratrol is added, and the solution was sonicated to produce a primary emulsion. This emulsion was slowly injected into bovine serum albumin solution to enhance the stability of the nanoparticles. This was again sonicated to ensure that the resveratrol would bind to and be encapsulated by the PLGA molecules. To disperse this emulsion, 15 ml of deionized water was added and stirred magnetically for hours. It was centrifuged and the supernatant removed, leaving behind the nanoparticles. These were washed with deionized water. The nanoparticles were lyophilized by vacuum freeze drying under negative 50 degrees Celsius and stored at 4 degrees Celsius. These could be stored for up to 6 months with negligible change to the encapsulation efficiency and drug loading. To prove this, the nanoparticles were periodically assayed at 0, 1, 3, and 6 months, looking at the particle size, morphology, and encapsulation efficiency. The particle size was determined using dynamic light scattering, and morphology was measured by field emission scanning electron microscope. The release of resveratrol from the PLGA nanoparticles was determined by imitating digestion conditions in gastric, intestinal, and intravenous fluid. Resveratrol PLGA nanoparticles were dispensed into 3 dialysis bags. The bags were each put into a buffer solution at pH levels 1.2, 6.8, and 7.4 to simulate gastric, intestinal, and intravenous fluid, respectively. The in vitro release was performed by continuous shaking at 37 degrees Celsius, again to mimic the conditions of the human body. 1 ml was sampled at predetermined intervals at 2, 8, and 24 hours, and every 24 hours after this up until 216 hours. The concentration of resveratrol released from the nanoparticles was measured spectrophotometrically using a microplate reader. The total amount of resveratrol released over the 216-hour period was calculated. Non-alcoholic fatty liver disease was modeled using oleic acid. Oleic acid is a naturally occurring fatty acid and was used in this experiment to induce hepatic steatosis to be used as a non-alcoholic fatty liver disease model. The intracellular accumulation of triglycerides, or fats, was tested by oil red O staining, as seen in figures 7 below. Oil red O staining stains the triglyceride content within the sample, in this case giving a visual representation of the severity of non-alcoholic fatty liver disease. In order to detect and quantify the effect of resveratrol and resveratrol PLGA nanoparticles on fat accumulated post-oleic acid induction, the triglyceride kit A110-1 was used. Post-induction, cells were incubated with free resveratrol and resveratrol PLGA nanoparticles for 24 hours. Accumulated triglyceride levels in cells were measured according to the instructions of the kit to determine the degree of fat accumulation. To detect and quantify the effect of resveratrol and resveratrol PLGA nanoparticles on lipolysis, which is the breakdown of lipids and fats, post-oleic acid induction, a glycerol kit was used. Again, post-induction, the liver cells were incubated with resveratrol and resveratrol PLGA nanoparticles for 24 hours. The glycerol content was measured according to the instructions of the glycerone enzyme kit, E1002, to determine the degree of lipolysis. So now that we're familiar with the method, let's take a look at the results. As seen in figure 8a, both resveratrol and resveratrol PLGA particles were proven to reduce the percentage of lipid accumulation significantly. Resveratrol PLGA nanoparticles, however, proved to have the greatest effect on reducing the fat content of the liver, having a reduction by 80.77%, whereas free resveratrol only reduced it by 71.01%. It's important to note that as the concentration of resveratrol and resveratrol PLGA nanoparticles increased, the percentage of lipid accumulation decreased. Figure 7 shows the oleic acid-induced hepatic steatosis stained through oil red O staining. The red staining represents lipid accumulation. Figure 7a shows the control HepG2 liver cells with the addition of different concentrations of oleic acid. 250 micromolars of oleic acid proved to be the optimal concentration, as higher doses cause high levels of cytotoxicity, and thus this concentration was used for the testing of resveratrol and resveratrol PLGA nanoparticles. Figure 7b shows the HepG2 cells induced with 250 micromolar of oleic acid with the addition of different concentrations of free resveratrol compared to a control. It is very apparent that as the quantity of the resveratrol increases, the red staining seen in the image dissipates, and hence it can be said that less lipid accumulation occurs. Figure C shows the HepG2 cells induced with 250 micromolar of oleic acid with the addition of different doses of resveratrol PLGA nanoparticles compared to a control. Again, as the concentration of resveratrol PLGA nanoparticles increases, the amount of lipid accumulation significantly decreases. Proved that resveratrol can prevent and treat non-alcoholic fatty liver disease in the early stages, and that PLGA nanoparticles enhance the drug's stability, solubility, and pharmacological potential.
Now we'll be talking about the discussion section of the article. The article had many limitations in terms of its long-term toxicity assessment, as it only ran the tests for toxicity on the cell levels for 4 hours, which is a not really substantial amount of time if you think about how long you would have these particles in your body for treatment, potentially be days or months. So this is a significant limitation in terms of its use in the treatment of NAFLD. The paper also had another limitation with the correction of errors. An error in figure 6 was corrected after the paper was released. This gives a possibility of more errors potentially within the area of results that could impact the analysis and conclusions made in the paper. And then we have speculation. There was speculation of whether pH affects oligomer uniformity and the release of RSV, as there were no experimental results to confirm these assumptions. This would require additional research in subsequent studies. And then we had the unclear number of replication of cell cultures for the multiple experiments in terms of whether or not they were used different cell cultures or sub-cell cultures for each test. This lack of clarity raises questions as it can affect the results of the test. If you use different cell cultures for each replication, you get different results, therefore limiting the reliability of the results. And now I'm moving on to the strengths of the paper. So I've had proved hypothesis from the authors in terms of their results showing that nanotechnology helps enhance the bioactivity of RSV, as well as having substantiated conclusions based on the results of decreased triglyceride accumulation, increased glycerol release in the hepatocytes, and decreased hepatocellular proliferation. These changes are required for the treatment of the effects of NAFLD. And then it also had reliable outcomes measured by quantitative assays with quality control and reliable associations with them, as having machines measure the results of reactions, remove bias as the scientists can't influence the results, and then reducing human error as well, giving these experimental results more reliability. Another strength is the fact that the data was free of any outliers, which indicated valid results, as there was no indication of mistakes made in the experimental procedure. Now we're moving on to the control group. There was multiple control groups present for each test, which validated the assay's performance to industry standards. This ensured reliability and quality control was maintained. And the authors also had appropriate statistical analysis in terms of using a pH value of 0.05 and 0.01 to determine the significant data points, as well as using t-tests and one-way analysis of variance to check the group's mean difference from one another. This added validity to the results as well. We also had prior studies that the article confirmed in their outcomes with the improved uptake of RSV in the body, as the previous study showed that nanoparticles were helpful with the stability of various drugs for different treatments. And additionally, we had no current research for the stability of RSV or its medium for delivery. This allowed the paper to explore a section within the molecular biology field that had yet to be investigated, allowing other scientists to see the results of this, and then further thinking on areas that they could improve on what the study had looked at. So in conclusion, we had an optimized method of preparing PLGA nanocarriers as 50 milligrams of PLGA, 9 milligrams of RSV, 10 milliliters of BSA solution, and 4 hours of stirring time. That was determined by the different experimental outcomes. They were also able to obtain 97.25% of encapsulation efficiency, as well as 14.9% of drug loading. So they changed the method, so you could change the methods and further experiment with future studies that could potentially increase that efficiency. And then the paper also had decreased lipid accumulation and increased glycerides with RSV nanoparticles from the result of cellular testing with the RSV compared to the RSV nanoparticles. This demonstrated the sustained and slow release process of less than 70% release RSV from the RSV NP particles after 216 hours that showed nanoparticles help with the stability of the drug when transporting through the intestinal tract. So most of it will be released within the liver cells and increase the effectiveness of RSV. The paper was also able to solve existing problems of poor aqueous solubility, chemical instability, and the metabolism of RSV within the intestinal tract, as RSV within the cells of the liver was shown to increase. The paper was able to show potential for the NAFLD treatment with the promoted lipolysis and reduced hypercellular proliferation, which was a major effect of NAFLD. This study was a key piece of research for the field of molecular biology in terms of demonstrating nanotechnology's improvement of drugs in treating various diseases, and the future studies can explore the different compounds that RSV can be encapsulated to enhance bioactivity and how nanotechnology can potentially be used with different drugs for different treatments of diseases in the future. As this is the first study for RVS treatment with nanoparticles, there's a lot more research to be done before it can be applied to the human body in the future.
Okay, and here are our references. Thank you so much for listening.